Good morning. Am I audible, visible? Hello, everyone. Am I audible, visible? Vanakkam Pasangla. Welcome to Unacademy NEET English. I'm your biology educator, Ambika Sharma. See, that's what I've learned. Hi, everyone. Welcome to Unacademy NEET English. I'm your biology educator, Ambika Sharma. And again, we are going to continue molecular basis of inheritance. And today we are going to talk about the experiments, the search for the genetic material. Okay, that's what we are going to cover. So, welcome to the session. So, tell me, am I perfectly audible and visible? Happy morning, everyone. Happy morning. See, I've learned something in Tamil. That's Vanakkam Pasangla. Pasangla means kids. That's what I've learned. So, welcome, welcome, welcome. Happy morning. So, let's continue the class, guys. But before that, let me appreciate them. Very good, Trisha. Very good, bache. Yesterday, right, I asked you few questions and you people typed the answer here in the comment section. Very good. I'm so proud of you all. Thank you for being so consistent. Thank you for taking your homework seriously. Thank you so much for revising the things properly. Right, I'm so proud of you. So here you guys can see, so yesterday I was asking about that melting point. So it depends upon cytosine and guanine because of that triple hydrogen bonding. Very good, bache. So the difference in A type DNA, Z type DNA is given here. Very good, Suhashini. Very good, Vidisha. Very good, Akanksha, Priyanka, and user. Okay, we will put the video of chapter 8, that is cell. Fine. Good morning. Good morning. So, guys, if you want to join any of our batch, right? Just say your HSP sir is taking chemistry mind maps, okay? And here you guys can see the link for our Avengers 2.0 batch. So, you can be the part of this batch. What you have to do, you just need to click the link there, add your number, your email. After paying fees, you will be the part of our batch. So, still, bache, 60% of the syllabus, you will get the classes for that syllabus live and the rest will be in the recorded format. And don't worry, whatever you have missed in the revision round, you will get the classes for that as well. Okay. Okay, so join the batch as soon as possible. Right now, again, after coupon code, it is just at a price of 4,687. And here you are even going to get the test as well and doubt sessions and the revision classes as well. Okay, so let's go. Let's start the chapter, that is molecular basis of inheritance. So what you have revised so far? Suhashini, Dinesh, Akanksha, CM, Madhu, Jayashri, tell me quickly in the chat section, everyone, and hit the like button. And if you are new to our channel, do subscribe our channel. So quickly tell me. Yes, quickly tell me what you have revised so far. Adenine is going to pair with? Adenine is going to pair with? Very good, Shiva. Adenine is going to pair with? Quickly answer it in the chat section. Very easy question. Thymine, of course. Cytosine is going to pair with guanine. So as per Chargaff's rules, write down the points of Chargaff's rules. Chargaff's rule. Write down the points of Chargaff's rule. Quick. Quick, quick, quick. Write down the points of Chargaff's rule. Very good. Purines is equals to pyrimidine. So adenine and thymine. Adenine, the amount of adenine is equals to the amount of thymine. The amount of cytosine is equals to the amount of guanine. Moreover, the amount of purines is equals to amount of the pyrimidines, right, bache? But when you talk about a particular species, when you consider a species, that A plus T to G plus C ratio, that is right, constant in a particular species, but it is variable in different species. Okay, that's what we have studied so far. So now let's continue, bache, and we are going to talk about the search for the genetic material, that it is DNA. Now you already know that somewhere in 1869, right, your Frederick Mesher. In 1869, Frederick Mesher, he discovered that nucleus in the pus cell. Okay, he called it as nuclein. But that quest for the genetic material, that which substance, which macromolecule is actually a genetic material, it started very late. Okay, when you look at this paragraph, right, this is what we are going to discuss today, the search for genetic material. Today we will be talking about the experiments, and according to me, this is the easiest part of this chapter because it is also easy, interesting, and moreover, for sure you will get question from today's lecture. Okay, you can get one question, even two questions from today's lecture. So please take the notes as well. Okay, so see, as I said, the discovery of nuclein by Mesher and the proposition for principles of inheritance by Mendel were almost at the same time. Right, at the same time, you know, Mendel explained about that principles and the Mesher about that nuclein, right? But that the DNA act as a genetic material, it took long to be discovered. Fine, it took long to be discovered, as I said, by 1926. Here, years are important, right? In this particular chapter, these years, they are important. So by 1926, the quest to determine the mechanism for genetic inheritance had reached the molecule level. Imagine in 1926, right? In 1869, Frederick Mesher discovered this genetic material, but discovered this DNA, but that it is a genetic material, right? It took so long. By 1926, they got to know about it. Imagine. Okay, so here you guys can see previous discoveries by Gregor Mendel, Walter Sutton, Thomas Hunt Morgan, and numerous other scientists had narrowed the search to the chromosome located in the nucleus. You know it very well. But the question of what molecule was actually the genetic material, it needs to be answered, and this is what we are going to discuss today. See, I don't think we are going very slow because in the morning that is the slot I have available. And see, we always used to say one thing, that when it comes to your syllabus, right, we are going to complete it, we know that, but we are not going to rush. Fine, we are not going to rush because we have some learners, those who are in class 12th. Okay, they want a proper revision for boards as well, for NEET as well, and these are the most important chapters. Right, these are the most important chapters. So in the morning, the chapters that we are taking, for that I'm taking 5 lectures, 6 lectures, but in the evening, in one part or two part, we are finishing the chapter. So I think speed is fine. Okay, but if you are finding it slow, you can increase the speed of the video. Okay, done. So now we need to talk about what? We need to talk about the transforming principle first. So you should know about the, yes, you should know about the scientist name as well. So what are we going to cover today? Transforming principle. So bache, transforming principle was given by your Griffith. Okay, who gave this principle? Who gave this principle? It was given by Frederick Griffith in 1928. In 1928. Now, he conducted an experiment. I'll directly come to the point here. Okay, he conducted an experiment, right? For his experiment, he used Streptococcus pneumoniae. This is a bacteria, of course. It is Streptococcus. Coccus means spherical shaped bacteria. Okay, so Streptococcus pneumoniae, also known as Diplococcus pneumoniae, right? So he used this bacteria, but this bacteria is having 2 strains. So he performed the experiment by using that 2 strain. One is your S strain, another is your R strain. How many of you know about it? One is your S strain and another is your R strain. So S means smooth here. Okay, R means rough here. Fine, these are the 2 different strains of the Streptococcus bacteria that Frederick Griffith has used. Clear, bache? Clear, bache? So it is also known as S3 strain. Fine, and this is your R2. Like in the case of bacteria, you know that they are prokaryotes. Okay, okay, if you link it with, yes, bache, if you link it with the biodiversity, so you know that when we talk about the biodiversity, when we talk about the number of species, we do not even consider prokaryotes. Do you know about this, Subhashini, Nandini? Yes, CM? Tell me. Tell me. Bache, there is no full video of molecular basis of inheritance, Shri. We need to take the one shot yet. So see, we have started this channel few months back. So we need to deliver you the quality content first. Let me teach you people in detail and then we will just start with the one shots. Fine. Okay, so what are we discussing? The transforming principle given by Frederick Mischer in 1928. As I said, the year is important. So he has used the bacteria named Streptococcus pneumoniae or Diplococcus. So it is the one which can cause pneumonia and it is having 2 strains. So I was linking this part to the biodiversity. So in the biodiversity, we have studied that when we talk about the number of species, we only consider eukaryotes. We do not talk about the prokaryotes. Why is it so? Why? Because in the case of prokaryotes, even if you talk about a bacteria, it is having different strains. Even if you check at their composition, you will find the differences. We do not have that techniques, right, that we can study each and every bacteria and its property. Are you getting it? So that's why I'm saying that when it comes to the bacteria, they have different type of strains because of the differences in, you know, even some few compounds. Okay, so S strain is there, R strain is there. So S stands for smooth, R stands for rough. Now what is the meaning here? See, S strain is your virulent. Virulent means it is pathogenic. Pathogenic means it is the one which is going to cause the pneumonia. And this R strain is non-virulent. Okay, what's that? It is non-virulent. So it is non-virulent means it is non-pathogenic, means it is not the one that can cause pneumonia. Okay, that can cause pneumonia. These are 2 different strains. So why do we say that one is smooth and another is rough? Why? Let me tell you this point. See, when you talk about the bacteria, you know that it is having the coverings, like it is having no doubt cell membrane. Fine. Even bacteria is having the cell wall, and outside the cell wall you are going to find the cell coat. These are the coverings of a bacteria. The cell coat is actually glycocalyx. Okay, it can be slimy, it can be solid as well. So smooth strain is the one in which bacteria is capsulated, in which the bacteria is having a capsule, right? That outermost cell coat, the capsule is present here. What is present here? That capsule is present here, right? It is made up of polysaccharide and it is the one which is responsible for virulence. It is the one which is responsible for being pathogenic. Are you getting it? Are you getting it? So these are the 2 different strains. This will cause pneumonia, this will not. Now, obviously, we need mice for it, isn't it? For conducting the experiments, scientists, they took what? They took mice. So what they did, see, this is the thing. It is given in NCERT. Let's use it. So when it was S strain, and when S strain, it was injected into mice. Mice die. Why? Because of pneumonia. Fine. Because of pneumonia. Clear? Then when it was R strain, it was injected into mice. Mice live. Why? Because pneumonia is not there. It is understood, right? It is understood that when S strain was injected into the mice, mice died because of pneumonia. But when R strain was injected, mice survived because pneumonia was not there. Now, they are going to, Griffith was able to kill bacteria by heating them. Then what Griffith did? He heated up that bacteria. Firstly, that S strain of bacteria. Okay, so he observed that when it is the heat killed S strain, right, when it is what? When it is heat killed S strain, bacteria injected into mice did not kill them, right? See, when S strain is heat killed, it is not killing the bacteria, mice. Why? Because bache, this bacteria is dead, right? That capsule is destroyed. Are you getting it? Are you getting it? Okay, next is, now see, this is something, you know, game changing. For an example, just a minute. Your heat killed S strain, what's that? Your heat killed S strain, what's that? It is non-pathogenic. Yes, it's non-pathogenic. And your rough strain, R strain, it is also non-pathogenic. It is also non-pathogenic, yes or no? So if I will insert both of them in the mice, mice should survive or die? Tell me. Mice should survive or die? Tell me. Obviously, mice is going to survive, yes or no? Heat killed S strain plus R strain when they were injected into the mice, because both are non-pathogenic, right? So mice should die, but Mice should survive, but what happened? Mice died, bache. Mice died, right? So then it was a question mark for Griffith, that how is it possible? Because heat killed S strain is also non-pathogenic, R strain is also non-pathogenic, then why that mice died? Clear, bache? Clear? So he came to the transforming principle then, right? What he concluded that there is something in that heat killed smooth strain bacteria. There is something in that heat killed smooth strain bacteria that transformed R strain into the virulent one, okay, into the pathogenic one, okay. So that's why we got this principle of transforming principle. Are you getting it? So that's what he concluded, that heat killed smooth strain transformed R strain into the virulent one. But at that point they were not sure that it is DNA. He just gave some transforming principle, that there is one substance in one particular bacteria that can transform into the another one. But it was not clear cut, right? It was not that they didn't get that clear-cut answer that it is the DNA. Okay, so this is what you guys can read here, that he concluded that R strain bacteria had somehow been transformed by heat killed S strain. So some transforming principle transferred from heat killed S strain had enabled R strain to synthesize that smooth polysaccharide coat, and then it became virulent. So bache, question can also come from this part, that what is making this S strain virulent? It is that polysaccharide coat. It is that outermost capsule. Is that clear, bache? Sangeeta, is that clear? Yes. Okay, so it is very easy experiment, isn't it? That's a very easy experiment. Okay, so you should remember the scientist name as well. So now the next is, bache, your every MacLeod. They conducted tests, they performed that biochemical analysis as well. This is also another important point, right? See, next is the biochemical characterization of transforming principle. So bache, question can come in your paper. They can ask you this thing as well, that who performed biochemical, who did that biochemical characterization of the transforming principle? So the answer is your Avery, MacLeod and McCarthy. Clear, bache? Scientist names can be asked in paper. This is important. Fine, this is important. Clear, bache? So prior to the work of Oswald Avery, Colin MacLeod, Macklin McCarthy, the genetic material was thought to be a protein. Means before that, they all were thinking that proteins are the genetic material. Proteins are the one which will pass from one generation to another, right? But their work determined the biochemical nature of that transforming principle in the Griffith's experiment. So this is what they did, bache. This is what they did. Like, I will see, look at this. They performed many experiments, right? That's the kind of final result. Just look at it. They took that S cells in a liquid medium, they centrifuged it, right? Then they killed it with heat, right? So they recover the S filtrate. This is the complete experiment. Just look at this, Suman. This is the complete experiment. What they did, S cells, they took that S cells in a liquid medium, they centrifuged it, right? They centrifuged it. So S cells spun to the bottom of the tube, they killed it with heat, and finally they got the filtrate. So basically they got the filtrate of that S strain of the bacteria. First point clear? Yes, bache. First point clear, that this is what they get. Now, see, in this filtrate, firstly they treated that filtrate with deoxyribonuclease. Right, deoxyribonuclease. So bache, here the question is deoxyribonuclease. Can you tell me the role of this enzyme? They are talking about this DNAs, and even in NCERT they are also asking you the difference in DNA and DNAs. See, it's very simple. DNA is your genetic material. Okay, that's what we need to conclude. But you know that it's a polynucleotide chain. Fine, it's a double helical, double stranded, right, polynucleotide chain. But when you are using the word DNAs, it is an enzyme, right? It is an enzyme. It is your deoxyribonucleases. right? It is your deoxyribonucleases. So what is nucleases? Nucleases is the category of the enzyme that hydrolyze your nucleic acid, right? That eat your genetic material, your nucleic acid, okay? So nucleases are of two types. One is your RNAs, another is your DNAs. So DNA is the one that will digest genetic material, that will digest DNA. RNase is the one that will digest RNA. So DNase is an enzyme which is going to hydrolyze, which is going to digest nucleic acid DNA. Clear, Subhashini? So now if I'm taking this filtrate, okay, now if I am taking this filtrate, see, they were not aware that it is the DNA which is the genetic material, but we know that, isn't it? We know that, isn't it? Isn't it? Sangeeta Bache, he will take the classes soon. Okay. There is no point of spamming in biology class for physics class. Both the subjects are important, no? So kindly focus here. He will take the classes soon. He is stuck in some family emergency if I'm not wrong. So he'll take the classes. So kindly do not spam here. Fine. So what am I saying, Bache? So say, imagine there is a filtrate. I'm taking a part of this filtrate and I am treating this filtrate with your DNAs. So what is going to happen here in this filtrate? Your DNA will be digested, yes or no? Yes, Priyanka, your DNA will be digested. If in the filtrate I am adding the DNAs, obviously it is going to digest the DNA. So of course, this filtrate will not help in the transformation of our cells. Our cells will remain non-pathogenic only. Yes, Nandini, our cells will remain non-pathogenic only. No transformation is there. When they treated the filtrate with RNAs, because it is going to digest your RNA. Again, when they perform this transforming experiment, see, what are they saying? Transformation occurred. Because now they have treated it with RNAs. So RNAs is going to digest the RNA only. Remaining DNA, protein, carbohydrate, lipids, all are present here. Okay, so they were like, right, like we should start it from this side. Let's say if they treated the filtrate with any enzyme that is digesting carbohydrate, obviously transformation will still occur. If they are going to treat the filtrate with the enzyme that is a protease, that is going to digest the protein, again the transformation will occur. Again the R cell will become S cell. Again the R cell, R bacteria will become the virulent one. Okay, but when they perform the experiment with the DNAs, they observe, then the transformation is not there. So it means if DNA is getting digested, if DNA is not there in the filtrate of that, in that filtrate, it means that it is the DNA which is the genetic material. Yes, Suman? Yes, Suman? So this experiment, this biochemical characterization was performed by, again I'm repeating, scientist's name is important, Avery, MacLeod and McCarthy. Clear, bache? So they purified biochemicals like proteins, DNA, RNA from the heat killed S cell, right? And they checked that which could transform live R cells into the S cells. So they discovered that DNA alone from S bacteria caused R bacteria to become transformed. Clear, bache? Clear, bache? So they also discovered that protein digesting enzyme protease, RNA digesting enzyme RNase did not affect transformation, right? But digestion with DNAs inhibit the transformation. So now you know what is this DNAs. Clear, bache? Clear, bache? So this is the complete experiment. Fine. Fine, so they concluded it is a DNA which is the heredity material, but not all biologists were convinced. है ना? We scientists, they are also like relatives. They are knowing, they will not be convinced. You have to give them something solid. Okay, so that final experiment which actually proved that it is the DNA which is our genetic material, it is your transduction principle. Again, another MCQ. Right, but from each and every line of this chapter, the question can come. Fine, the question can come, and whenever I'm saying that this is a previous year question, this is a previous year question, please solve such questions, bache. Solve such questions, and you're going to find them in your last 10 years PYQs or even in 15 years PYQs. Fine, fine. So this is about your transforming principle. So the final experiment which actually proved that it is the DNA which is the genetic material, it was performed by your, yes, it was your transduction experiment, also known as blending experiment, and it was performed by Hershey and Chase. That's very important. It was performed by Hershey and Chase in 1952. So Alfred Hershey, Martha Chase, 1952. So this is transduction experiment or the blending experiment having 3 steps. Clear, bache? Having 3 steps. Is that clear? Yes. Is that clear? So 3 steps are infection, blending, centrifugation. That's what we need to discuss. Okay, see, these are the 3 steps of this experiment. This diagram is from NCERT na? These are the 3 steps of this experiment. Infection, blending, centrifugation. Infection, blending, centrifugation. So that's what we need to do. But before that, yes, you have to understand few things. Okay, you really need to understand few things. So let's go for it. See, for this experiment, what Hershey and Chase, they did. Yes. So what they did, let me tell you. First of all, they experimented on, they took bacteriophage. Okay. And of course, the world famous bacteria, E. coli, isn't it? Isn't it? In all the experiments, this is what we are going to get. Your E. coli, right? Light bache, Suman, Sangeeta? है ना? It is the E. coli, the world famous bacteria. The world famous bacteria. So these 2 things were used by Hershey and Chase. Now the point is, E. coli, as I said, it's a bacteria, of course, right? It's a gram-negative bacteria. It's a gram-negative bacteria. So anyone in the class who can tell me what is the meaning of being gram-negative? Yes, why am I saying it's a gram-negative bacteria? Because it is not getting stained with the gram stain. Okay, Kristen Gram has formed one stain and gram-negative bacteria is not getting stained with it. Why? Because it is not having that thick polysaccharide coat. This is what you need to remember. Okay, now this is the bacteriophage, lambda bacteriophage. Okay, what is it? It is a bacteriophage. Now, what do you know about the bacteriophage? Can you tell me? Okay, it is the T2 phage actually, not the lambda phage. It is a T2 phage. It is the bacteriophage. What is the meaning of a bacteriophage? But a bacteriophage, it is a hyperparasite. Why am I saying it's a hyperparasite? Because it is parasitic on bacteria. See, it's a virus actually. What is it? This T2 phage is what? It's a virus. It's a DNA virus. Okay, it's a DNA virus. Its genetic material is double-stranded DNA. When you talk about the bacteriophages, they are the viruses. Okay, so these are the viruses which are parasitic on bacteria. These are the viruses which are parasitic on bacteria. So any parasite which is parasitic on a parasite, right? Any parasite which is parasitic on a parasite, for that parasite, we use the word hyperparasite. Right? Like, let's say I am a bacteria, I am parasitic. You are a virus, you are parasitic. You are parasitic on me, right? I am eating my host, you are eating me. So you will be the hyperparasite. You will be the hyperparasite. Clear, bache? Clear, bache? So bacteriophage is a virus that is parasitic on bacteria. Okay, so we call it as hyperparasite also. So it's a DNA virus. Okay, it's a DNA virus. Clear, bache? In biological classification also, we have covered the structure of the bacteriophage. So when you talk about this bacteriophage, question can come from this its diagram. Okay, in previous year papers, there is a question from the diagram of the bacteriophage. So if you will see this, do you know what is this head? You know, this is the head. Here you will see the tail fibers. Okay, this is the head, but actually it's a protein coat. It is made up of what? It is made up of proteins. Fine. It is made up of what? It is made up of proteins. Can you see that? This diagram of bacteriophage is important. Okay, the diagram of bacteriophage is important. It's a virus. Fine, it's a virus. Clear, bache? Yes. Is it clear? So here what can you see? It is a protein coat. Let me draw this. That's a protein coat. You know that in the viruses we have that protein coat, we call it as capsid, isn't it? We call it as capsid. That's your DNA. This part is the tail here, bache. This part is the tail here. Here you will see the sheath in this tail. Here you will see the collar. Here you will see the collar in this tail, and these are some tail fibers. Okay, these are what? These are the tail fibers. So that's what bacteriophage is. Okay, that's what bacteriophage is. Now, see, ultimately in this experiment, this bacteriophage is going to infect your E. coli. In this experiment, this bacteriophage is going to infect the E. coli. So they conducted this experiment, but in two sets, like two experiments were performed simultaneously. Okay, two experiments were performed simultaneously. In experiment number 1, they used radioactive sulfur S35 isotope, right? Radioactive sulfur S35 isotope. Clear, bache? S35 isotope. And in another experiment, in experiment number 2, they also use that radioactive isotope. Again, they use this radioactive isotope, but it was P32. What was it? It was P32. Another important question. Now, why P32? Because this phosphorus is present in DNA. This phosphorus is present in DNA. Actually, this experiment was conducted to check whether it is the protein which is the genetic material or it is the DNA which is the genetic material. So when it comes to the DNA is having phosphorus. So they used radioactive phosphorus, okay, so that it can get incorporated into DNA structure. Okay, so that it can get incorporated into DNA structure. Second is S35. Why S35? Because it will be the part of this protein coat, right? It will be the part of this protein coat. So radioactive sulfur, if they are using, it will get incorporated here into this protein coat, into this capsid, and the phosphorus 32 will get incorporated with the DNA. That's what you need to keep in your mind. Okay, that's what you need to keep in your mind. And moreover, I hope you remember there is one trick for that. So it is Chief Minister, sir. It is Chief Minister, sir. That is your cysteine and methionine. Your cysteine and methionine, these are the sulfur containing amino acids. What are they? These are sulfur containing amino acids, and such amino acids are present here and they are making the protein coat. So this part clear? This part clear? So as I said, this experiment will be performed in 3 steps, right? In how many steps? In 3 steps. Infection, blending, centrifugation. Do you know the meaning here? That what is the meaning of this infection, blending and centrifugation? Yes, bache? Yes, do you know that? See, in the first step, what will they do? Let's say this is a culture in which bacteria is there and your radioactive labeled bacteriophages are there. Okay, so in the very first step, they will make sure that bacteriophage is infecting your bacteria. First step clear, bache Trisha? First step clear? So both type of labeled phages means radioactive S35 as well, radioactive P32 labeled DNA as well. So they will take 2 different cultures, right? And they will make sure that in first step, these bacteriophages, they start infecting the bacteria. First step clear? First step clear? Then comes the blending, right? Then comes the blending. So in the blending, in a blender, because now in first step, bacteriophage has infected the bacteria. In the very first step, bacteriophage has infected the bacteria. Like see, this is your bacteria's DNA and this is your radioactive protein coat containing bacteriophage, and here See this. Okay, okay, see this. So this is the first step, which is known as infection. Clear? So second step is blending. So they are going to take this in a blender. They will agitate this mixture so that bacteriophage and bacteria, they can break the content, they can break the contact. Okay, so bacterial cells are going to be agitated in a blender to break the contact between virus and bacteria. Okay, okay bache, so what is going to happen in this step? The bacterial cells were agitated. They were agitated in a blender to break the contact in between bacteriophage and bacteria. Why is it so? Why is it so? Because in the very first step, bache, right, when there is the infection, obviously this is the nature of, right, do you know that this is the nature of your bacteriophage, that when it infects the bacteria, it passes its genetic material, it passes its DNA to that bacteria, it incorporates its DNA to the host's DNA. That is the nature of this virus. You know, that's how the viruses work. They are non-living outside the living body. When they are not getting their host, they are just proteins. They are just inert crystallizable proteins. Okay, that's what a virus is. It is a connecting link in between non-living and living organisms. So when a virus, it gets a living body, it gets a host, then virus uses that host's machinery and divide and make multiple copies, right, and make multiple copies. So here in first step, this is the duty. This is what this bacteriophage or the virus is going to do. It will pass its genetic material to this E. coli so that it can incorporate its DNA with E. coli's DNA, and it can use E. coli's machinery to make multiple copies. Clear, students? Is that clear? So this is the second step, blending, and when there is a third step, that is centrifugation. These are the three steps. So you should know about the sequence as well. Right, you should know about the sequence as well. So virus particles were separated from bacteria. Here in centrifuge, virus particles they'll be separated from bacteria. Fine, they will be separated from bacteria. So that is what centrifugation is. Clear, bache? Centrifugation is, so they are going to obtain the filtrate. So just have a look. Okay, so see, so now you know that when this is the protein coat which is radioactive, so obviously radioactivity will not pass within the E. coli, yes or no? That much you know. At that time they were not aware, but because of their experiment, at least you know that it is the DNA which is the genetic material. So obviously radioactivity which is there in DNA, that will pass to the bacteria. That will pass to the bacteria. Now here, because protein coat is radioactive, so that radioactivity will not enter the cell. Okay, so when they will in centrifuge, when they will separate virus and the bacterial, virus particles and the bacterial particles, so what will they see? That radioactivity is in supernatant. Where is that radioactivity? It is there in that supernatant. Like, let's imagine, so this part is the, see, upper, this portion, which, this part which comes above, it is actually the supernatant. Okay, it is actually the supernatant. Fine, it is actually the supernatant. Is that clear? Yes, right? So this is just the pellet. This part is the pellet. So here they will see, when it is S35, they will see that supernatant contains the radioactivity. It is, and this part, let me tell you this thing, bache, don't get confused. Actually, this pellet is showing the, this is heavy one, na? So it is showing the bacterial content. It is showing the bacterial content, and this part is showing your viral content. Clear, bache? So in viral content, in the supernatant, you will see the radioactivity, not here in the pellet, not here in the bacterial content. Are you getting it? Right? But when it is the radioactive phosphorus, because that radioactivity, because DNA It is the DNA which will pass to the next generation. So radioactivity entered the cell. So they will see that radioactivity is in palate, and palate is showing what? Palate is showing the bacterial content. So there is no radioactivity in supernatant. There is no radioactivity in supernatant, right? So what they concluded, that it is the phosphorus 32 which is passing to the bacteria. Means it is the phosphorus 32 which is incorporated in DNA. So it is the DNA which is passing to the next generation. It is the DNA which is causing the transformation. It is the DNA which is the genetic material. Is that clear? Yes, is that clear? So this is all about the experiments, bache, right? I have added another chart as well. So first is showing the labeling of this bacteriophage. Clear, bache, right? So now bacteriophage, in first step, bacteriophage will get labeled. So infection is there. So DNA labeled phage is going to infect unlabeled bacteria. Then blender agitation is there, bache, and finally the lysis will be there. Okay. Okay, so the notes will be provided to you in the Telegram group. Our official Telegram group is Unacademy NEET English, right? In the description box, link for that Telegram group is given. You guys can join it. Okay, so I will provide you the notes there. So this is all about this experiment. So radioactivity detected in supernatant, which is showing viral content, and this one is showing what? The bacterial content. Is that clear? Is that clear? So that is what your transduction experiment, right, or the blending experiment is saying. But do you know why do we call it as transduction experiment? Yes, do you know why do we call it as transduction experiment? But yeah, whenever any virus is used for transferring the genetic material, whenever any virus is used for transferring the genetic material, that is known as transduction. Like in the case of bacteria, we talk about the transformation, we talk about the transduction, we talk about the conjugation as well. So when will be the conjugation? When there is a tube, right? That tube will pass the genetic material from one bacteria to another. You know, here there will be the role of plasmids, F plasmid, fertility plasmid. But here what are we discussing, students? We are just talking about the virus. The virus is going to pass its genetic material. Virus is used to transfer the genetic material. So that's why this is known as transduction. And when will it be transforming? When any bacteria is picking up the DNA from the surrounding, it will be the transformation then. Clear? Clear? So this is all about the experiments, right, bache? This is all about the experiments. So the next topic is the properties of the genetic material, then RNA word, and finally the semi-conservative nature of DNA. That's what we need to start. So if there is any doubt, do let me know. A question will definitely come. Even if you are preparing for boards, in CBSE board, question used to come from this part. Done? But why do we say it is the DNA which is our genetic material? Or what property should a genetic material have? What properties? Very simple it is. If it is a genetic material, it should have the property to replicate. Okay? 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 So anything which is a genetic material, it should have the property of self-replication, first point. Then after replication, it should have property that it can pass that information. First of all, it should store information. It should know how to replicate that. Then it should know how to pass that genetic material or the information from one generation to another. These are some basic things. Right? These are some basic things. And moreover, when we say DNA versus RNA, here comes the structural stability. Like if you talk about the DNA, no doubt, bache, when you relate it with the evolution, okay, when you relate it with what? When you relate it with the evolution. So in the evolution, when we talk about that chemogeny, the chemical evolution, we used to say that monomers, right? First of all, you know that atoms, molecules, elements, compounds, inorganic compounds, organic compounds came to this planet, right? And then like they formed, basically they formed in water. And then we used to say that simple, or you can say that simple organic molecules having low molecular weight, they formed first, then polymers, they formed first. That is how we discuss. That is how we discuss. Yes or no? Yes or no? So initially it is believed, and this is what RNA word concept says, right? Like basically scientists, they believe in two things. Some scientists say it is the RNA which came first. Some scientists believe that it is the protein which came first. So RNA, those who believe that it is the RNA which came first, now RNA word concept, and one is protein word concept. See, why do we say that it is the RNA that came first? RNA, bache, it is a nucleic acid. We know it very well. It can store genetic information. It can pass that genetic information to the next generation. Moreover, this RNA can itself act as an enzyme also. Remember ribozyme. Remember ribozyme. So initially it was very important, right? When we were talking about the evolution, right? It was very important. If we want a living structure, so obviously it is important to have something which can take a control of it, which can take a control of that machinery, which can help it to grow and to divide, right? Like if you relate it with evolution, I hope you know about the concepts of coservates. I hope you know about the concept of microspheres. Okay, Fox has given the microspheres. So yeah, microspheres they are. So basically we talk about some structures now which were cell-like. They were not living, Nandini, but they were cell-like, and slowly prokaryotes formed. Why? Because when this genetic material formed, because it has started taking the hold of everything. Okay, so many scientists, they believe in this concept of RNA word, that it is the RNA which came first, and it can behave like the nucleic acid as well as the enzyme. But some, they still believe that it is the protein which came first, the enzymes they came, and then further the evolution proceeded. Clear? This is RNA word concept. And when you compare RNA, so okay, if we have RNA, then why do we need DNA? Of course, with evolution, we moved from simplicity to complexity. Yes, we moved from simplicity to complexity. That's how we move. That's how we proceed. Of course, yes, right. That's how we move. That's how we proceed. We know it very well, isn't it? We know it very well, isn't it? Isn't it? Yes. So when it comes to the RNA is your single-stranded, bache. RNA is your single-stranded. Its sugar is ribose. It is having uracil. They make it more reactive, and because this RNA is single-stranded, it is quite unstable because it is more reactive. Being single-stranded, it is more reactive, so it makes it unstable. Moreover, uracil and ribose having that extra OH group makes it more reactive. It's like that. So we want a stable genetic material. Then with evolution from RNA, right, we got this DNA. Because DNA being double-stranded, it's less reactive. It is having thymine. It is having deoxyribose sugar, which is, helping it more to become the stable. Okay. So that's how you relate this part of NCERT. Okay. Right. So see, from the foregoing discussion, it is clear that the debate between protein versus DNA as genetic material was unequally resolved from Hershey and Chase experiment. This is what they used to ask in NEET examination, right? So Hershey and Chase experiment, right? It resolved that conflict, that whether it is protein or the DNA as the genetic material. So it is the DNA which is the genetic material. Clear, bache? So you know that even RNA is also the genetic material in some viruses, and we used to call such viruses as retroviruses. Like in plants, TMV, tobacco mosaic virus. It is also a RNA virus. Clear, bache? Your QB bacteriophage is also the RNA virus. So these examples are important. Please make the notes, bache. Please make the notes. So it is actually the DNA which is the predominant genetic material. I will tell you why. So please have a look. And see, they are saying, can you recall the two chemical differences between RNA and DNA? I'm pretty sure you know that. Yes, Niranjana, what's your doubt? I'm pretty sure you know that, isn't it? So a molecule that can act as a genetic material, it should be able to generate its replica. I told you. It should have the property to replicate itself, and you know that your DNA is having that. Remember, in S phase of cell cycle, the DNA replication will be there. Then it should chemically and structurally be stable. Your DNA is. Okay. It should provide the scope for slow changes. Right? It should have property that it can get mutated, but not at a faster rate. RNA being single-stranded, it can also get mutated, but at a faster rate. We need the genetic material in which mutations are there, but at a slow rate. Why? Because mutations are sudden changes, right? So slow such changes are required for the variations, but not at a fast rate. Okay, not at a fast rate. Clear, bache? So it should be able to express itself in the form of Mendelian characters, like Mendel was explaining, no? Right? Character is having two variant traits. One is controlling, like basically, you know that how it works. It is in pair and all. So this is what they are saying, that it should be able to express itself in the form of Mendelian characters. Clear, Niranjana? Mendelian characters. Clear? Clear? Sure. So if one examines each requirement one by one, right, because of rule of base pairing and complementarity, both the nucleic acids are having ability to direct their duplications. Even your RNA can form its duplicate DNA. Why are we discussing the rule of complementarity here? It's very simple. If we have the DNA, right, if we have one strand of DNA, if we know A is present here, so we know that we should add T here. If G is here, we know we should add C here. So this can help in the duplication, right? This can help in the duplication. Clear, bache? Clear, bache? But other molecules like your proteins, they do not fulfill this criteria. So for sure, it is your RNA, DNA, these nucleic acids which are the genetic material. Is that clear? Is that clear? And as I said, the genetic material should be stable enough not to change with different stages of life, age, or with change in physiology of the organism. Clear, bache? Clear? Clear? So this stability was even evident in Griffith's transforming principle, that heat which killed bacteria, it was not able to destroy the genetic material. Clear, bache? Clear? So see, this point is very, very, very important. Please mark it. Please mark it. Okay. Further, 2-OH group present at every nucleotide in RNA is a reactive group. I told you na, why ribose sugar? Why ribose sugar was replaced by deoxyribose sugar in DNA? Why? Because that extra 2-OH group in the ribose sugar was making it more reactive. Okay, it was making it more reactive. It was making that RNA labile and easily degradable. Okay, easily degradable. So that is something very important. Okay, and RNA, you know that it is also catalytic. That is why it is more reactive. Okay, okay. So DNA chemically is less reactive and structurally more stable when compared to RNA. That's why DNA is a better genetic material. So this is what we have discussed here. Okay, done, bache? Done? Or see, the presence of thymine, as I said, at the place of uracil also confers additional stability to the DNA. Fine, bache? So both DNA and RNA are able to mutate, but RNA mutate at a faster rate. Why? Because it is single-stranded. Okay, okay, and that is the reason whenever you talk about any virus which is having RNA as the genetic material, in that case it's very difficult to form the vaccine. Okay, like when we were talking about the coronavirus, when we were talking about that COVID-19, you know that what was happening, because that is a RNA virus. It was mutating at a very fast rate. So scientists, they were not, like, once they were getting that structure, but again it was getting mutated. So in that case, it is very difficult to form the vaccine if we are targeting the genetic material. So in that case, no doubt we can focus on receptor, but it will also change now because your RNA is mutating. Okay. Okay. So that's why in the case of RNA viruses, it becomes difficult to form the medicines as well. Fine. Fine. So RNA genome, it is having shorter lifespan, mutate and evolve faster. Fine, bache. So RNA can directly code for the synthesis of proteins, hence can easily express the characters. So DNA, it is dependent on RNA for synthesis of proteins. So you know that that is the flow of information. That is the central dogma of information. Fine, fine. So both DNA and RNA can work as genetic material, but it is the DNA which is more stable, preferred for storage of genetic information. So for the transmission of genetic information, RNA is better. And even that all that protein and their evolution, it revolve around your RNA. Hanji, which is Selva, please tell me what's your doubt. I will answer the question, okay? I will answer the question. Tell me quickly. Tell me quickly. So students, up to this part all clear? Sure? Sure? Are you getting it? See, we are covering the topic in detail as well. We are reading the NCERT as well. So it is more like the NCERT series. Okay? Okay, so kindly use this and revise it again and again. Now we have completed NCERT as well. We have completed the topic as well. So after the class, every day, please practice MCQs. Fine, please practice MCQs. Right? So on YouTube, now that's the basically issue, because it is a kind of one-way interaction. I'm trying to make it two-way, but you know, you people are giving test or not, I don't know about it. So if you are the part of my batch, I know it, right? You will give test after every 15 days. Fine. So that is quite easy for me. Then I can check your progress as well. Okay. So see, RNA word. Okay. So which one is the first genetic material? Right, this is what we have to discuss. So it is the RNA which was the first genetic material. Okay, okay. So there is now enough evidence to suggest that essential life processes like metabolism, translation, splicing, everything evolved around RNA. So it is also a genetic material as well as catalyst. You know that. Then we will call it as ribozyme. Fine. Then we will call it as ribozyme. So, but RNA being a catalyst is reactive, so it is unstable. So that's why DNA has evolved from RNA with chemical modifications, that is making it more stable. Fine, that is making it more stable. So this is all about the RNA word concept. And next is the mode of replication. Next is the mode of replication. Okay, that how this DNA will get replicated. That's what I said now. The main property of the genetic material is that it should have the capability to get replicated. Okay, that it should have the capability to get duplicated. This is what I just said, right, bache? Right, bache? So now here, this is what we are going to discuss. So when you are talking about the models of replication, we have majorly 3 models of replication. Okay, when you talk about the models of replication or modes of replication, so we have 3. One is conservative, one is semi-conservative, another is dispersive type of. Okay, so when it is conservative, the word is explaining everything. Here, this word is explaining everything. Conservative mode of DNA replication. But you see, you know that DNA is having the information it is. And this complementarity can help us to understand that in a better way. Like if I will open up this DNA, okay, right, I know here there are certain bases present. And I know they are going to follow the rule of complementarity. So let's say if I have the T here, I know I have to add A. If I have the T here, I know I have to add A. If there is C, G. So in that way, we can form a strand complementary to this parental strand. Complementary to this parental strand? Of course, yes. So when it is the conservative mode of DNA replication, right, when it is the conservative mode of DNA replication, so what is happening? Both the parental strands, they are available for copying the information. They are acting like a template. They are acting like a template. But later on, what is happening, bache? This new strand, because it is also complementary, it is joining like this and parental strand is joining like this, right? So parental strand will remain intact as such. Okay, no doubt after this DNA replication, without any doubt, you will get 2 daughter strands. But here one is totally parental types, like parental type is conserved here, and next this will be that new strand, new DNA strand. Clear, bache? So it is conservative. So parental strands will open up to copying the information, but at the end of the replication, both the parental strands will re-anneal and the new daughter template strand, both new daughter strands, they join and they form this. Okay, this is conservative. When it is dispersive, it's very different type of bache. Like in the daughter DNA, you know what will you see? That some part of daughter DNA some part of daughter DNA is parental, some part is new, like this, like this. That's dispersive, like this. And when it is the semi-conservative, it is the one that we need to discuss, bache. When it is semi-conservative mode of DNA replication, in this case, what is happening? Parental strands, they will open, bache. They will behave like the template. Why do we use the word template? Template means you are actually using that part to make something else, right? Like, let's say I'm giving you one task. Okay, I'm telling you that you have to make a file. Okay, you have to make the biology practical file. So I will show you one file. Okay, this is how your file should look like. So accordingly, you are going to work. So now in the case of this DNA, this is double-stranded DNA. When I try to, so I will try to open it up. Will tell you that in DNA application. So when I will open it up, it's strands. I will get these strands. So these strands are having information and they are helping me to synthesize the new strand. So that's why I'm using the word template for it. That's why I'm using the word template for it. So complementary to it, new bases will be added. Then after DNA replication, what is happening? When you will see the daughter strands, here one strand is parental types, another strand is the newly synthesized. One strand is parental type, you can see this, another strand is the newly synthesized strand. So see, in the conservative, parental DNA will remain as such, new daughter DNA will form. In semi-conservative, one strand is parental type, another is the newly synthesized, and it is the dispersive model. It is the dispersive model. Is that clear? What is it? It is the dispersive model. Clear, bache? So while proposing the double helical structure of Watson and Crick, proposing the double helical structure for DNA, Watson and Crick, they had immediately proposed a scheme for application of DNA. Okay, okay. So this is their exact quote: to has not escaped our notice that the specific pairing we have postulated immediately suggests a possible coping mechanism for the genetic material. So basically what they were trying to say, because they were studying the structure, they have noticed that complementarity there. So they were like, obviously it is also telling us its replication way as well. So they just proposed it, right? They didn't provide any proof for it. They just proposed it. But when it comes to the proof that yes, it is the semi-conservative mode of DNA replication, it was provided by, yes, can you tell me? Stahl. You know about the scientist? Do you know about the scientist? Michelson Stahl, he provided that. Okay, so scheme suggested that two strands would separate, act as a template for the synthesis of new strand. After the completion of replication, each DNA molecule would have one parental and one newly synthesized strand. So it is the semi-conservative mode of DNA replication. Okay, okay. So it was proposed by Watson and Crick. So while answering this question in final paper, please check the language as well. So proposed by Watson and Crick, but experimental proved. See, it is now proven that DNA replicates semi-conservatively. It was first shown in E. coli, right? And in plants also. So Michelson and Stahl, right? Matthew Michelson and Franklin Stahl, they performed the experiment and they explained it. Is that clear? Yes. They performed the experiment and they explained it. So, bache, this experiment and further DNA replication we will discuss in the next class. Okay. So, you please be consistent, revise everything properly. I have already posted the video for the complete strategy to score, right, 650 plus marks in 6 months. So, if you have started now, please go and check that video. Okay, go and check that video. I will provide you the PDF as well once the chapter is over, because I will provide you the complete PDF. Okay, so do revise it. Done, bache? Do revise it. Fine. So now if you have any doubt, please tell me. Bache Ahmed, just visit our channel. You will find the video. Don't worry. Thank you. Bache, Vinu, I will not say that I will go very fast, because this chapter, it needs attention. It needs your patience as well. right? If you're doing it for the first time, that's my request. Please go slow, because once you are done, once you understand the concept, then you just need to revise it quickly. There are certain points that you need to recall again and again, and the topic will be over. Fine. And then the topic will be over. So thank you so much. Stay tuned. We'll see you tomorrow. Tomorrow we do not have class at 10. Tomorrow we have lecture at 6, and we'll be covering body fluid and circulation. Okay, that's what we are going to discuss. Body fluid and circulation. That is our next one shot. Emma, do check that video. It will answer all your questions. Okay. So guys, now you know what you have to do. I'll wait for your comments in the comment section. Do let me know you like the session or not. Stay tuned. Thank you.